software programs that we use at Active Motif to analyze sequencing data begin with a software called BCL to FASTQ that is published by Illumina for Illumina sequencing data. This generates FASTQ files for a project after a sequencing run that we then use a software called Burroughs-Wheeler Aligner, or BWA, to perform our actual alignment to a reference genome. From there, we have our aligned reads, and we can perform the next step of an analysis, which is called peak calling. And so for peak calling, it depends on the factor that we're profiling. And so for things that have a narrow signal spectrum, we use a software called Max2 to perform peak calling, where for broader peak signals and histone marks, we'll use a software called Sicer to perform this peak calling step. And so that gives us information that is in more than just the reads that align to a particular place, but also a measure of their signal as compared to the background of the rest of the genome. And so after we have our peaks, this really becomes the main unit of our analysis that we can then transform to perform visualizations in things like the UCSC genome browser, IGV, IGB, or can have as input into a suite of R packages that we use for downstream differential comparisons and analyses. The quality control process for bioinformatic analysis at Active Motif first begins with an assessment of sort of base quality scores and adaptive contamination through a software called FASTQ Screen or FASTQC. From there, we use a multi-species sequence alignment tool to investigate for contamination of reads from another species like mycoplasma or things in a xenograft model. Beyond that, once we get the go-ahead from those checks, we will align our data and then can get a lot more, sort of sequence-centric, data-centric measures of quality like alignment rate, duplication rate, FRIP, or fraction of reads and peaks, and other various measures. And so we use those to say, okay, well, is this data usable for subsequent downstream analysis? If it doesn't pass those checks, then it's important for us to begin a dialogue with not only the lab, but also researchers whose project this is to see that we can confirm that this data is of high quality and what they're looking for their project. At Active Motif, when we're building our philosophy for what a project should contain, a bioinformatic analysis, we first look to the literature. So we like to sort of have an ear to the ground for how people are publishing in that field and what tools they're using for their projects. Beyond that, we also take information from different consortia or organizations that sort of have skin in the game for how this data should be analyzed so that it's easy when a researcher has their data in hand from us that they can easily compare to what else is out there and know that that data has been treated and processed in the same way. After those, something that we really value is feedback from customers or researchers that we work with. And so in opportunities where we have time to meet prior to an analysis beginning, we like to make sure that everybody is on the same sort of field expectation-wise for what we can deliver, as well as that the analysis that we're designing for their project ultimately meets their goals for the biological question that this project was intended for. In cases where we get opportunities to meet after the project has been delivered, it's really important for us to know that this is a lot of data that we're sort of throwing at you guys, and so where do you look in these tables and files that we've handed you that sort of contain the information to ask those specific questions? Oftentimes, this is the first time that researchers are seeing this kind of actual data in hand, and so we want to make sure that we don't just drop everything afterwards, and we continue to support the project after it leaves our hands and goes into yours.